Welcome to Innovations. I'm Ed Begley Jr. In the past, the lack of understanding and knowledge surrounding the healthcare community caused many to be misdiagnosed and mistreated. Today, advancements in science and technology are helping to better educate both physicians and patients on the growing options that are becoming available to patients around the globe.
Flow cytometry has become an essential tool in our scientific and medical fields today.
The core principles of flow cytometry begin with monoclonal antibodies, which allows scientists to know that they're going to measure one particular aspect of a cell and fluorescent probes that bind to these monoclonal antibodies. And so when you then shoot these fluorescent probes with light, they'll fluoresce. So you will know quantifiably how much of a particular antigen was present. And so you can measure, say, one particular aspect of a cell on many, many, many cells, or the more sophisticated the cytometer you have, you can start measuring more and more different aspects of these cells.
You think of a lamppost in a park, and you imagine that lamppost is on at night. Well, we can all imagine that that lamppost shines light in a very specific area. Flow cytometry does better than that. When you use flow cytometry as an analytical technique, you are looking beyond the light of the lamppost, and it allows you to take big facets of medically important data into decision-making in a simultaneous and real-time way.
It is highly specific. It can measure millions of cells simultaneously, and it can allow you to look at multiple populations in the same sample. You hear often about chemotherapeutic drugs that really wipe out the bone marrow. You kill the cancer but kill the patient. Well, this allows us to fine-tune those aspects. We can look at what is happening not only at our target, but with everything surrounding that target.
If you remember, there was a big breakout in the Human Genome Project. We mapped the whole human genome, and now we know that every A, C, T, G pair that was going on, but you don't hear a whole lot about it because it's hard now to figure out how that system actually processes and leads downstream to outcomes. Flow is different. Flow is happening right now, and so you're able to say, if I have treated this test subject with the drug, what are the outcomes, what are the results? And so this really lends itself well to drug discovery. And then once you get farther into the process, you can actually start saying, all right, well, which drugs now are going to be safe? Which drugs are going to have greater efficacy? You can test these against millions of cells, and you can do it very quickly.
Having a simple instrument available to many will speed up diagnostics. Speeding up diagnostics means also initiating treatment faster. So it means better outcome for the patients, less cost for the healthcare system, and overall better health for everyone.
With more than 80 combined years of expertise in flow techniques, the Pennsylvania-based company FlowMetric is an expert in flow cytometry.
FlowMetric is a niche CRO that was founded in the spring of 2010. Flow cytometry is actionable analytics. In essence, it's a way of allowing a company developing a medicine to go towards success faster by the answers we provide, but also to fail quicker. And what that really means is being able to pull up stakes quickly when something is not going to be a successful medicine. So it allows the company to save money by making actionable decisions early on.
Flow cytometry assays are essential for the development of modern medicines and will play a key role in the introduction of personalized medicines used for cancer patients.
Right now, we're working with the breast cancer group at Penn to develop an assay to detect something called disseminated tumor cells in breast cancer. These are the cells that cause relapse breast cancer, and today, there's almost no treatment for relapse breast cancer. What happens is these cells, which are a very, very, very small population, go hide out in the bone marrow. They've tried doing genomics, but the signal to noise ratio is too low and they can't detect it. So we're able to find, using the specificity we have with flow cytometry, 1 in 10 million cells. We can combine that now with genomics, because the other aspect of flow cytometry, there's two parts. It's analytical, and you can identify a cell, but you can also sort it out and do it. So we can sort out that single cell and do single cell genomics and get very specialized precision medicine for precision diagnostics. So that means we can ask a lot more questions. What it also means is we have a lot of data.
FlowJo, the software that my company makes, is the market leader. And so it's the software that people have learned how to analyze flow data on in general. Additionally, FlowJo now can be put on a server so that we can really crank through these large data sets using a supercomputer.
A combination of flow cytometry and creativity, like you see here at FlowMetric, that's what's going to make a difference, and that's what's going to really be innovative in the future. But what's unique about the approach that FlowMetric is taking is they're taking that technology, they're addressing unmet needs that are identified by the end customer with creative ways and ingenious ways to make a difference in those settings.
Standard cytometers, commercial cytometers, are essentially confined in centralized labs, big labs where the samples are going to the instrument, and they are made to process a lot of those samples and batch them together. And so that's why it's taking a few days to get your results back from a centralized facility. Having a smaller instrument and a more robust one, of course, because you're going to move it around, is really taking the instrument to the samples, to the people, instead of the other way around.
One of our most recent objectives at FlowMetric has been to marry good diagnostic assay development with flexibility and mobility. And from that, we have derived the MoPod as a mobile point of diagnostic with the understanding, expectation, and hope that it will be a very useful way of rapidly deploying laboratory medicine in the field. Deploying it anywhere in the world at a moment's notice, for instance, to monitor infectious disease outbreaks, and then here at home as a way of bringing laboratory medicine to the patient in doctors' offices, in pharmacies, and ultimately in patient homes. Flow cytometry is relevant for everyone on this planet. It not only is a gateway to understanding the human condition, but it's also the gateway to ultimately understanding our relationship to the planet itself. Flow cytometry can be used to monitor global warming. It can be used to monitor the outbreak of new pathogens. So ultimately, it's something that we all will care about or should care about. It's a great growth industry.
For more information, visit www.flowmetric.com.